This module is on the strategies for non-clinical immunogenicity assessment. I am Helen Haggarty, and I will be leading you through this module. While taking this module, you will learn about the value of non-clinical immunogenicity assessment and regulatory expectations, testing strategies with regards to assays and study design and key considerations to those, and how to interpret the impact of immunogenicity in non-clinical studies. I will end the presentation with a case study illustrating the immunogenicity assessment and the interpretation of the impact of immunogenicity on a non-clinical study. So let's refresh on your knowledge of the consequences of immunogenicity and whether it be in humans or in animals. Anti-drug antibodies may be generated that have little to no impact on your study, but they can also have consequences that can be significant. They can have an impact on efficacy of the biotherapeutic, either by altering its PK and or its pharmacodynamic activity. This can happen in a couple of different ways. You can have clearing antibodies, which can be generated that bind to the molecule and increase its rate of clearance. The formation of these antibodies and drug complexes are recognized by the RES system and then are removed from the systemic circulation. This results in a decrease in the distribution of the drug to its target. On the other hand, for some proteins, the anti-drug antibodies actually decrease the clearance rate. And in this case, they are called sustaining antibodies. This has been seen with some hormones and cytokines, which often have very short half-life of hours. And when bound to the anti-drug antibody, which has a much longer half-life of days, can keep it in the circulation longer via the FcRn-mediated drug recirculation. You can also have neutralizing antibodies, and these are antibodies that bind to or near the target, the binding of the target, such that it can no longer bind to its endogenous target, and this leads to reduced biologic activity. In addition to impacts on efficacy, you can have an impact on safety, and this can manifest in many different ways. They can be general adverse immune-mediated toxicities, such as hypersensitivity reactions, that can be acute in nature, or localized injection reactions, infusion reactions, or those that are more delayed, such as cell-mediated reactions or immune complex disease. In addition, cross-reactive antibodies that not only bind and neutralize the drug, but also bind and neutralize the biological function of an endogenous protein counterpart can be generated. If the protein they are neutralizing mediates a unique biologic function with a critical physiological role, its neutralization can result in a deficiency syndrome or worsen the disease it's intending to treat.